Now it's time to discuss some experiments on photosynthesis. These experiments tell you the conditions or the requirements that are there for photosynthesis. So what is the basic criteria when we discuss these experiments? You need to just remember that during photosynthesis, glucose is made, the food is in the form of glucose. Now this glucose, it is stored in the form of starch. Now for this, what do we need to do? We need to de-starch the plant. How do we do de-starching? We keep the green plant in darkness for around 24 to 72 hours. So how this happens? When you place the plant in darkness, the starch, that is the stored food in plants, it is used up in the cellular activities. And after this, no new starch is being formed. So we can say that by keeping the plant in darkness for this amount of time, we can have a de-starched potted plant. Now what do we need to do next? If we talk about the starch test, we have iodine solution that tests the presence of starch. We take the leaf from that potted plant which was kept in darkness, remember, and that leaf, it is kept in boiling water. So what happens when we keep it in boiling water? This ensures two major things. The first one being that when we keep the leaf in boiling water, this actually denatures enzymes. What it does, it denatures enzyme. Now when the denaturing of enzymes is there, then there will be no new chemical change, right? So when the enzymes are denatured, no new chemical change will take place. Secondly, then this ensures permeability of the iodine solution. Since when we boil the leaf in boiling water, this is going to lead to the, the rupture of the cell wall, the rupture of the cell tissues. So this ensures more permeability of the iodine solution. So what is this iodine solution? How can you make this iodine solution? It's very important to understand. You'll take 100 ml of water, and in this 100 ml of water, just put 0.3 gram of iodine and 0.5 gram of potassium iodide. So when you keep both these two things in 100 ml of water, you say that you have made an iodine solution. Now when you put this iodine solution on the leaf, if it turns into blue-black colored component, you will see a blue-black color, then this confirms that starch is present. If it does not turn blue-black, if the leaf parts does not turn blue-black, it means starch is absent. So what happens? This iodine solution, it reacts with the starch present in the leaf and it forms a complex which is blue-black in color. And if starch is not present, then definitely no complex will be formed and the color will remain as it is. The iodine solution's color will remain as it is. So what do we need to do next? When we have, we have that leaf from the de-starched potted plant, we'll keep it in boiling water. Then we're going to take that leaf out, we're going to put it in alcohol, right, in a test tube containing alcohol, and we do not directly, take alcohol solution and keep it on the flame. Why so? Because alcohol is highly flammable. It can catch fire easily. So we take the help of double boiling method. What do? We actually put in water in the beaker, and in that boiling water we put in the test tube that is containing alcohol, right? And in this test tube we have that leaf that we have already boiled in boiling water. So what happens? You will see that after some time the chlorophyll pigment, which is green in color, which provides green color to the leaves, it goes into the alcohol, right, since it is soluble in alcohol. So we have this leaf now, we keep that leaf in a petri plate, we put the iodine solution. If it turns blue-black, it means starch is present. If it does not turn blue-black, means starch is absent. So let's see how we actually do this starch test. We need a leaf, we need boiling water, we need methylated spirit or alcohol, iodine solution and a spirit lamp. So what do we do first? We take water, remember, in water we put that leaf from which plant? The plant that has been kept in darkness for this much hours. Then we boil that leaf. This is going to ensure the denaturing of enzymes and permeability by the rupture of cell tissue. Then we take that leaf in a test tube containing alcohol or methylated spirit. And then we are going to put this test tube in the beaker which is containing boiling water. Why? Because we cannot directly heat it, because alcohol is quite flammable, it can catch fire and lead to accidents in the lab. So we're going to put this test tube in the beaker containing boiling water. So as you can see, what is going to happen? I told you the chlorophyll pigment that is there, which causes the green color of the leaves, it is going to solubilize in alcohol. Why? Because like dissolves like, right? So you will see a color change in the alcohol. What sort of color change you will see? You will see that the alcohol that is there in the test tube, it turns green. Why? Because all the chlorophyll that was there in the leaves, it has got into this alcohol, right? So now we are going to take that leaf out and keep it in a petri plate. You will now put which solution for starch test? That is the iodine solution. You know how to make it, right? Then when we put the drops of iodine solution into it, you can see if it does not change color, it means starch is absent. And if after some time it changes into blue black color, it means the starch is present. So this is how we do the starch test. This is how the starch test is performed, by using this iodine solution, right? So I hope that the procedure for the experiments that we conduct during photosynthesis is clear to you. This was the basic procedure, right, which we follow for destarching a plant, which we follow for the starch test. So these are the basics which you need to know when we discuss some experiments on photosynthesis.